Today I'm going to be talking about using blue waters to do some design of peptides for bioelectronic applications. So the particular series of peptides that we're interested in is what we term the DXXX series. It has these aromatic cores in the center and aspartic acid termini on both ends. And then the design part is you can kind of just fill in these side chains with whatever amino acids you like. Why is this an interesting set of peptides? Well, in high pH, the aspartic acid termini are deprotonated and negatively charged, so these small proteins tend to repel one another. But when the pH is lowered, they become protonated, which leads to a minimization of electrostatic repulsion and large-scale self-assembly. And if the resulting aggregates are sufficiently ordered in some sense, that is, if there's sufficient overlap between the pi orbitals of these aromatics, then the electrons can delocalize down the backbone, and there will be some kind of fluorescent properties, which makes these peptides nice candidates for bioelectronics, such as organic photovoltaic cells, organic light-emitting diodes, or biocompatible pH sensors. Now, what I was interested in, as I said, I'm kind of interested in the design. So what we wanted to look at was, can we come up with rational design principles that will lead to good aggregative properties in some sense? And what we've previously showed computationally is that the DXXX series demonstrates this kind of hierarchical assembly, where first we form what we term optical clusters, where there's some kind of overlap between those cores. And then the optical clusters interact via side chain, and core side chain interactions to form what we term contact clusters. And these contact clusters don't have any guarantee of proximity between the aromatic cores. So one of the things that I kind of have in mind going into this is that I'm probably interested in increasing the growth rate of the optical clusters, for example. Now, we run into a problem pretty quickly, which is probably one that's familiar to a lot of people here. We start out, we want to understand these things at a molecular level. We have experimental collaborators who can do experiments. Our feasible length and timescales for atomistic molecular dynamics are somewhere down here. Our experimental length and timescales are somewhere up here. And we're really interested in designing these things in a rational sense to have good aggregative properties. But if we're confined to length and timescales down here, then we're not really going to be seeing much aggregation at all. And certainly, even we can probably push this up a little bit, maybe a couple orders of magnitude if we use sufficient computational resources, but it's still going to become prohibitively computationally expensive at some point. So at that point, what we did was instead, we went to a couple of different, more coarse-grained models. The one I'm mostly going to be talking about today is this Patchy model that's really, we're losing some resolution, but we're really getting up towards experimental length and timescales, and with a good computational infrastructure, we can even scan over the different parameters of this model in order to understand how they affect the aggregative properties of the system. So in this model, this very simple model, each one of these peptides is a set of squishy Leonard-Jones beads that are rigidly constrained together. We represent each side chain as just a single bead that has some size and some stickiness, and that's most what I'm going to be focusing on today. Then we represent the cores as these blue beads decorated with sticky green beads on the top and bottom because the cores tend to be flat, and so that's kind of getting at the more directional cofacial interactions of the cores. And then, as I said, we're able to, come on, oh, it's not going to do the animation, sorry. Adobe Reader, we don't have Adobe Reader. But rest assured, there's some nice aggregation happening here. And like I said, we can do a nice parameter sweep to understand the different interactions. And first thing we can do is we can do a nice sanity check and see that, in fact, we are really kind of bridging between the atomistic or the higher resolution and the experimental length and timescales. So right here, this is a snapshot of our Patchy one of our Patchy systems after 660 microseconds. You can see it's forming this kind of branched porous network that has fibrils with widths on the order of about 10 nanometers. And that's just exactly what we see experimentally. You form these kind of branched porous networks with fibrils on the orders of about 10 nanometers. And then if we look at the log of the correlation integral versus the log of distance, the important thing about this plot is that the slope of any of these lines at this length scale gives the fractal dimensionality of the system on this length scale. So all of these systems, irrespective of their different parameters, are basically two-dimensional on a large enough length scale. So that's why it doesn't matter what our parameters are, we're still going to see this experimental behavior because this is kind of baked into the rigid geometry of these peptides. But then on these shorter length scales, on length scales of about 10 nanometers, we see that we get a dimension between 1 and 1.8. And if we look, for example, at one of our higher resolution coarse grain models, for a specific system, we have a fractal dimensionality of 1.5. So that's encouraging. And we're going to go on to understand how are these different interaction parameters actually controlling this morphology. And so what I'm showing you here is a series of snapshots of different parameters. And you can see that the microscopic morphology is different. These three have pretty non-sticky side chains. This has pretty sticky side chains. And red here is the cores. This blue is the side chains. And you can see for the sticky side chains, there's a fair bit of blue interacting with blue compared to over here where you're getting more red interacting with red. So as you get stickier, you're seeing that these things are more disordered and we're getting more side chain interactions. If we look at these three, this middle one has quite large side chains. These two have pretty small side chains, and you can see that for the smaller side chains, we're forming much more these kind of flat stacks and bundles compared to this kind of twisted fibril that we form for these large side chains. So to describe this a little bit more rigorously, if we look at the optical cluster growth rate with respect to the side chain stickiness, this is increasing side chain stickiness, and the side chain size, this is increasing side chain size, the first thing we see is that there's a pretty big effect of the side chain stickiness, as we might expect. As we make the side chains less sticky, like I just showed you, we're increasing the optical cluster growth rate. And we get the largest increase when we drop the stickiness below the stickiness of these cores. So essentially when we make side chain interactions more favorable than when we make side chain interactions less favorable than core interactions, we grow our optical clusters better, as we might have expected. If we look at the optical cluster growth with respect to the side chain size, we can see that we've got a kind of non-monotonic dependence happening there. Firstly, if we look at contact cluster growth with respect to side chain size, there is a very strong monotonic dependence, because as we increase the side chain size, we are increasing the collisional cross-section, which means we just have more collisions, so we're growing our contact clusters faster, which can lead to growing the optical clusters faster because you can have a contact cluster that then rearranges into an optical cluster. But on the other hand, we have fewer configurations available that these things can interact with where they are actually optical because of these steric constraints when the side chains get bulky enough. And that also affects the morphology, as I mentioned. That's why we're seeing, a twisted fibril for large side chains as opposed to these kind of flat sheets. And so it's really kind of there's these two competing interactions. So this leads us towards thinking about, well, okay, we know we want to have non-sticky side chains. We might want to have larger or smaller side chains. It kind of depends. But now what we can actually do is we can actually just identify optimal parameter sets given some objective functions. So what I'm showing you here is all 60 different parameter sets measured with two objective functions measured. On the x-axis, I'm measuring the optical cluster growth rate versus the contact cluster growth rate, which we'd like to be large, as I mentioned before. And along the y-axis, I'm measuring the fractal dimension on the order of the fibril width, which we'd like to be relatively small because then we're getting essentially you're getting a more one-dimensional system, you're getting more single interactions and therefore probably better stacking of the cores. And from this, so if we look at these, we want this to be large, we want this to be small. That means we want the points that are closest to the lower right-hand corner. That's these 2 points, and then these 4 points that are within error bars of these 2 points. And what we see is that all of these have, as we might expect, this turns out to be the least sticky. This is the smallest stickiness of the side chains that we measured. We get a range of different side chain sizes. And then this third parameter, which I'm not going to talk about too much today because it turns out there isn't really a strong dependence on this parameter, so we can't really come up with a very rational design principle for it yet. And so I'm not talking about it today. But we can go one step further with these optimal parameter sets because we can actually go ahead and map our higher resolution chemistries to this patchy model. So what I'm showing you here is that each one of these gray points is the side chain stickiness versus the side chain size of a particular chemistry. It's much, much cheaper to identify, to map that to this patchy model than it would be to go through and try to do the level of simulations that we've done in this much more coarse-grained space. And these 3 purple X's are the 3 optimal parameter sets in just the side chain stickiness and the side chain size. So they're all at 0.2 kT side chain stickiness or well depth. And then they have a range of side chain sizes. And we can pull out the closest chemistries next to these optimal parameter sets. And so for the smallest side chains, we pull out 3 chemistries made up of with glycines and alanines and their side chains that have not very sticky side chains and also not very large side chains. And then next to these kind of mid-sized side chains, we can pull out these 2 chemistries that have phenylalanine, methionine, and isoleucine residues in them. These are bulkier, but they still don't have very sticky side chains. And then it turns out we don't actually have any chemical candidates near this parameter set because in order to get side chains this large, we turned out to need to be significantly stickier. And we know that stickiness is probably going to be a pretty major factor in this, in getting us the aggregative properties that we think we want. Now, our next steps, so one thing we're pretty interested in doing is taking these chemistries as a starting point for an active learning procedure where we're maybe searching through our state space of chemistries, probably now at a higher resolution level, and basically doing some kind of smart learning procedure where we're trading off, have we looked in this space, do we actually expect this to be a good candidate? And so we might get some trajectory that looks like this, just for happenstance, for example. One thing I'm currently working on getting to work, but it's kind of in the beginning processes right now. So why blue waters for the system? Well, as I mentioned, this is a pretty large-scale problem. We were doing 300 simulations of 10,000 monomers apiece. Each one of those simulations required multiple GPU acceleration and produces 10 to 20 gigabytes of data to be analyzed. And not only did having access to blue waters provide us with access to the computational infrastructure that we needed in order to solve this problem, it also really provided us with an easily usable computational infrastructure because I'm a Blue Waters fellow, so we have all the resources from my point of contact and the entire NCSA team. I think something else that was really important for this project and is going to continue to be important going forward is access to the big data community that all of you are part of, because this is really a very interdisciplinary problem. One of the things that we are trying to work on going forward, for example, it's still a little bit stalled, is I've been talking about doing classical molecular dynamics, but I'm talking about optical properties. We still don't really have a rigorous understanding of the really fine-grained system and a really rigorous understanding of the electronic structure of these aggregates and how that changes as these aggregates grow and as they interact, as the peptides interact in different ways. So I think that going forward, knowing all of you, knowing various people in blue waters is going to be really important, because no one in our lab has experience with density functional theory, which we think is probably going to be the way forward on that side of the equation. So overall, what I've talked about here is coming up with this coarse-grained Patchy model that is recapitulating the series, the DXXX properties, but really bridging between the atomistic scale and the experimental scale. And I've come up with a couple of rational design principles. We know we need these non-sticky side chains. We know that we need to be very careful with the of the side chains in terms of what properties we're going to get out of the final system. I've also identified some optimal chemistries here. This is overall part of a multi-scale model for the rational peptide design. And as I mentioned, a couple of our future work, aspects of our future work, I said density functional theory. One other aspect of future work here that I think is really interesting is that we know that there is kinetic control of these aggregates. You can add, for example, an external flow on the system and change the properties of aggregation. And we know from earlier simulations that you can't see those effects until you get to the time and length scales that are reached by this Patchy model. And so that's another really pressing piece of future work that needs to be performed. At this point, I'd like to thank my advisor, Andy Ferguson, our collaborators in the Bill Wilson and Charles Schroeder labs, the Ferguson Lab Group, Blue Waters for my computational time and my funding as a graduate fellow and my support as a graduate fellow, also NERSC for some computational time, and various other funding sources.